eager to hear about nanowomen, aren't you? Well, in a not-so-distant future, an evil organization known only as Microcorp aimed to conquer the interplanetary system. Multi-millionaire nanoscientist Naomi Johnson, inspired by comic book superheroes Atom and Ant-Man, worked with her team of brilliant scientists. Nanowoman, the supersized superhero. When we last saw Nanowoman, she had discovered head Microcorp Agent Ace's plan to destroy the Chancellor with a cancerous growth guarded and sustained by microscopic Microcorp agents.
Look, we only have 2 days until the Universal Gala. If Microcorp has its way, the Chancellor will have a dramatic and mid-speech.
Honeymoon will ensue, and the system will be theirs for the taking.
Well, actually, guys, I've been studying the data from the doctor's note, and I've formulated the right cocktail for Herceptin, Pertuzumab, and Docetaxel. That should slow down the time and give you enough time to do your work.
If I get those Microcorp agents out of there, Johnson Industries can get the help the Chancellor needs.
Wait, how are you going to do that, though? They're everywhere. Once they get a whiff of Nanowoman, you're finished.
DNA Origami nanostructures!
What?
That's perfect!
That's the perfect way to get Nanowoman and the drug to the target location. Brilliant.
Slow down. Emmy here, remember? I fix the suits, I build the gadgets. I haven't touched biology since freshman year. This is all going way over my head.
Alright, so we'll break it down. DNA Origami nanostructures are basically made up of DNA strands that can respond to changes in the environment. These changes can be due to pH, it can be temperature, and it can be concentration. So you're probably wondering what DNA is, right?
The genetic material of the cell?
True, but that's an oversimplification. And if you come with me to the office, I'll explain it further for you. Okay. DNA consists of a double helix structure, which you can see here. And these structures have different nucleobases. And these bases are held together by hydrogen interactions and electrostatic interactions. And the double helix structure of the DNA was actually discovered by Watson and Crick using data that they got from Rosalind Franklin. And this is actually what the data looks like. It's famously known as 4051. And the X mark in the crystal structure is what helped them figure out that the DNA has a double helix structure. And that's basically how they figured out base pairing, what we now call Watson-Crick base pairing. So Watson and Crick base pairing commonly consists of, so the base pairs typically exist in pairs. The adenine and the thymine always bind together, and the guanine and the cytosine are always together. As you can see here, we have binding between the oxygen and the hydrogen, and also the hydrogen and the nitrogen. The other cool thing about Watson and Crick base pairing is that the DNA strands always run in parallel and they're complementary to each other. So for instance, in this one, on this strand on the left, we have it running from 3' to the 5' end, and on the flip side, it runs from the 5' to the 3' end. And we also see the AT and the GC base pairing that we talked about before. So there are basically different forms of DNA that can exist. There's the A form, there's the B form, and the Z form. And the B form of DNA is actually what all humans have in them. But the A form of the DNA is actually what helped Rosalind Franklin figure out the helix structure of the DNA.
Okay, but what do people actually use this for?
It's a good question. There have been several applications of DNA, and this has led to advances in fingerprinting, in cloning, in paternity tests, in gene mutations like in CRISPR, and most recently in DNA origami.
Oh, so that would explain how Modi can tell who the baby daddy is.
Yes, back to origami. DNA origami is made up of several DNA molecules that are mostly single-stranded and held together by short staple molecules. The length and shape of the DNA origami is determined in huge part by a DNA sequence, since DNA generally binds to a complementary sequence. Based on the arrangement of the double helix segments, hydrogen bonding and Watson-Crick pairs of DNA nucleotides can give rise to complex secondary structures. This folding technique, as you can see here, is one that is adopted by proteins in order to carry out specific functions.
Hence the origami part.
Right. Once folded, We can use them to make nanoscale tools, or in our case, drug delivery devices. That way, as the medication is moving through the body, healthy cells aren't negatively affected by potentially dangerous chemicals.
Once they reach their target location, they unfold. This could either be because of changes in pH or because of interactions with the target cell. And once they unfold, they release their contents to the current location.
Okay, so this plan could work, but how are you going to make them?
That's easy. We'll design single-stranded DNA molecule that will self-assemble based on its nucleotide composition. This DNA would be used as a scaffold to construct any shape that we want. After this event is completed, the DNA scaffold will be held in place using small strands of DNA known as staple DNA. The interaction between the staple DNA and the scaffolded DNA stabilizes the staple scaffold region over the secondary structure of the scaffold. The staple strands also act as intramolecular bridges between the scaffold, providing constraints that prevent the scaffold from folding onto undesirable structures.
This is similar to the DNA block method, which was developed by the Wilson Institute and basically relies on DNA short synthetic molecules being built up like Lego bricks. So I have a set of Legos, this can be a template stand, and you have the short synthetic molecules which bind D2 base pairing, and you can basically build up your structures like this. So this allows DNA to be preserved into different shapes, just like in DNA Origami.
Alright, back to origami. Once we create the DNA origami, we can code it so that it'll reach its target structure. That way, when it goes where it's supposed to go, boom, Houston, you landed.
We can pick from a variety of structures. Scientists have made simple box-like structures and have used scaffold strands of DNA to get more complex shapes.
Okay, so if I remember from class, DNA and RNA are actually structurally similar. Does that mean that there's such a thing as RNA origami?
Yeah, so as a matter of fact, that would be correct. RNA origami was actually developed in 2013, but the problem with RNA in general is that it's prone to degradation, so you can't really use it for a lot of things, unlike DNA.
So that explains why there's a whole lot of work done on it then.
Yep, was that helpful?
Yeah. But wait, last thing, I promise. It says here that DNA origami can be used for biosensors. What's that about?
As a matter of fact, that would be correct. Since the field of biosensing cuts across a diversity of fields, including medicine and feeding, there's a broad range of use for DNA origami. For instance, DNA origami has been used for protein and target molecule detection using DNA origami pliers. In the plier-shaped system, the nanostructure can transition distinctly between parallel, antiparallel, and X-shaped conformations, depending on non-covalent interactions.
Origamis can also be used as nanodevices for assembling biomolecules to perform pre-programmable tasks, and as a platform to design customizable biomachinery, using specific molecular structures such as protein-ligand binding interactions, the shape, or the profile of the binding sensor induces this change.
They're also very useful when used in nanofabrication to selectively functionalize the surface of nanoparticles and transfer molecular patterns on nano devices. While there's a lot of buzz going around DNA origami use, but a lot of research has been very fundamental so far.
True. However, the most exciting application so far is the field of drug delivery, like we're doing. Before we conclude on this subject, I want to bring to your awareness another application. Researchers at Harvard recently made nanorobots out of DNA origami. These robots even had hinges and could identify different proteins. The concept of DNA robots actually was just proved viable clinically in February of 2018.
Okay, so if DNA origami is so cool, why don't we see them everywhere?
You ask all the tough questions, don't you? Well, there's several reasons for that. The first one is cost. The cost of making DNA origami is really expensive because you have to buy DNA by the base pairs, and you need like 7,000 base pair for the template alone. And the second reason is that making the design of the DNA origami. You use a lot of materials to make these origamis, and they give very little yield, and so this presents problems for scale up. The third reason is that DNA origamis are typically unstable under physiological conditions, so like pH 7 and possibly room temperature, and so it's very hard for researchers to be able to conduct their experiments under normal conditions. And the last reason is that other origamis have been shown to like efficiently enter cells, like we use in drug delivery. The specific pathway by which that happens isn't quite known yet. These are some of the reasons why origamis aren't everywhere yet.
That makes sense.
Okay, so now that we know about DNA Origami and its applications, does everyone know what she's doing?
We're formulating the proper DNA sequence.
I'm selecting the coding for the proper structure. And I'm recalibrating the suits and gadgets. Still doing the same thing. Learned a lot though.
Good, then that means you're doing your job. And I'm going to let them know that Johnson Core's CEO is going to be at the Universal Gala. Now, ladies, let's move out.
What do you mean the Chancellor's still alive? I did all that undercover work and you still failed me? I guess we just have to move on to Plan B.